Hello everyone, I am Dr. Ruchira. Our today's topic is the neuromuscular junction and the mechanism of the muscle contraction. Muscle contraction mechanism. First of all, we have to know about what is the corticospinal tract. This corticospinal tract passes the impulse from the motor cortex, then travels through the internal capsule of posterior limb, internal capsule of posterior limb, and then to the midbrain, pons, and the medulla. It crosses in the medulla to the opposite side and go to the spinal cord. Go to the spinal cord. This is our spinal cord. Okay. So from here, from this step in the spinal cord, it is going in the two ways. 70% it's go to the inhibitory interneuron, and in 30% cases is it go through the anterior horn cells and again divided into two, that is A alpha and A delta. So this A alpha fibers are go to the causes the contraction of the extrafusal muscle fiber, extrafusal muscle fibers, and A gamma causes the contraction, A delta causes the contraction of the intrafusal. So basically, what is the extrafusal muscle fiber and the intrafusal muscle fiber? Suppose this is our muscle, this is our muscle. Okay, in this muscle there is a spindle present, muscle spindle present. If in the muscle spindle there is a presence of fibers, that fibers is the intrafusal fibers, and around the spindle means here there is a extrafusal muscle fibers. So what is the NM junction? That is neuromuscular junction. In the neuromuscular junction there is some steps. First is the opening of voltage gated sodium channel. Opening of voltage gated sodium channel. If the voltage gated sodium channel opens, so entry of the sodium channel occurs and action potential generates. Action potential generates. Then the release, there is the voltage gate action potential generates. Then there is a voltage gated calcium channels also gets open and calcium's calcium entry occur. Okay, then there is a then this calcium causes the movement of the vesicles, causes the movement of vesicles, and this vesicle movement in the vesicles uh, causes or releases the acetylcholine, acetylcholine, which is a neurotransmitter. This acetylcholine binds to the nicotinic receptor of the muscles. Nicotinic receptors of muscles. So, after the binding of the nicotinic receptors of muscles, the same three above steps are occur in the muscles. Suppose this is our muscle, and in this muscle there is a presence of a nicotinic receptors, and from the neuron the acetylcholine release and bind here on the receptor site. Then there is a action potential generates, and calcium entry is there. um, but in these two steps, um, in above we are already said that ki calcium channels are entry were done in the neuron uh, with the help of other stimulators, but in the muscle there is a storage site for the calcium, that is a SR. From SR, sarcoplasmic reticulum, the calcium is released and causes the moves the myofilaments, and this causes the muscle to contract. Muscle contraction occurs in this stage. Okay, so this is the flow chart, and very important flow chart for the exam point of view. So remember, first stage of the muscle neuromuscular junction is the opening of the voltage gated sodium channel. Then second step is the action potential occur. Third step is the voltage gated calcium channel entry, and fourth one is the movement of the vesicles due to calcium and the acetylcholine released. If in this above stage there is some inhibition occur due to any stimuli, then there is a some syndromes are caused if the auto antibodies to the voltage gated channel are destroyed. Means third step, inhibition of this third step occur, then uh, this is called as a LE syndrome. What is LE syndrome? Lambert Eaton syndrome occurs due to the inhibition of this stage. If there is a inhibition of a acetylcholinesterase occurs, then called as a OP compounds, organophosphorus compounds. And for this we have to give the treatment like a physostigmine drug, neostigmine drug, which blocks the acetylcholinesterase. Myasthenia gravis can occur. Um, when inhibition of a SNAP protein is there, so there is a no acetylcholine form and no contraction, and there is a flaccid paralysis we seen due to the Clostridium botulinum. What is the SNAP protein? SNAP protein is the SNAP proteins are of various name, that is a SNAP 25, snaptobrevin, snaptotagmine, uh, also a SNAP path. These proteins are helps to release the acetylcholine. If this is inhibited, SNAP proteins are inhibited, then there is a no release of acetylcholine. So there is a flaccid paralysis of a muscle occur. Flaccid paralysis occurs. What is flaccid paralysis? Means no contraction of the muscles are occur. if and this is due to the Clostridium botulinum. Clostridium botulinum. If there is a Clostridium tetani, Clostridium tetani was there, then there is a inhibition of a GABA or glycine inhibitory neurotransmitter. Then there is an increased acetylcholine, increased contraction, and causes the spastic paralysis. Causes the spastic paralysis. So we have to remember all this point regarding the neuromuscular junction, and the most common syndrome occurs due to the inhibition of the calcium channels. Then it is a Lambert-Eaton syndrome. Then release of acetylcholine inhibition, organophosphorus compound myasthenia gravis occurs if the SNAP protein are inhibited. Then flaccid paralysis, and it is due to Clostridium botulinum. If Clostridium tetani was there, then the spastic paralysis are occur. So if you are able to understand this mechanism, then like, share, and please comment below. It's so helpful for me. Thank you. Happy studying.